Greetings. Happy Tuesday, everyone. Thank you so much for joining us for another RRP-hosted research webinar. I'm Katrine Erickson. I'm the Executive Director of the RUNX1 Research Program. And for those of you who may not be familiar with RRP, we're a nonprofit organization dedicated to supporting individuals who have RUNX1 familial platelet disorder, which, as many of you know, is a genetic disease that significantly increases the risk of hematologic malignancies due to these germline RUNX1 mutations. And so at RRP, our #1 goal is to prevent heme malignancies in our patients and really improve their overall quality of life. So our work is inclusive of funding research, building and supporting our patient community, and facilitating collaboration across the many different stakeholders that are in our healthcare system, and doing all of this for that one goal of discovering a blood cancer interception and or prevention therapy. Our impact in research dollars has now reached over 12 million, and that's really only been possible thanks to the many co-funding partnerships we've had over the years. Just to share with all of you who are researchers, if you're conducting any kind of research that is RUNX1 FPD relevant, please do reach out to me. I'm always open to hearing what you're up to and whether or not you're interested in presenting at one of our webinars. So please don't hesitate to reach out to me. I'd love to hear from you. Also, if you're interested in applying for any one of our grant programs, please do keep yourself on our newsletter or in our distribution list so that you're aware of any new grant mechanisms. In fact, I'll share with you one grant mechanism that is available in a moment. And in fact, maybe I'll do that right now. So let me just go to one more page and then I'll introduce our speaker. So we do have an open RFA where we're supporting the clinical and genomic data set abstraction for patients that have already been consented for research. So we're interested in expanding the RUNX1 germline database that many of you are probably familiar with. We've now had that group expand and add on a REDCap portion to that database. So in connection with Variant Grid, we have REDCap. And so we're hoping to collect retrospective data from RUNX1 patients worldwide, and we're offering some monetary support to facilitate the labor-intensive portion of actually extracting that data and putting it into the database. So please check it out if you haven't already. And then now let me go back to, oops, forgive me, I just lost the presentation slide, but I think that's okay. So before I introduce our speaker, I do want to remind you that you can ask your questions throughout and use the Q&A icon to do that. So just type in your question and we can facilitate a Q&A session at the end for 10 to 15 minutes. And you're more than welcome to provide any comments in the comment icon as well throughout. All right, so without further ado, I'd like to introduce Dr. Hannah Carter, who is an associate professor in the School of Health Sciences at the University of California, San Diego. I was actually introduced to Dr. Carter via Dr. Alex Bick, who met her at a conference in Hawaii, a bioinformatics conference, I believe it was. And they met because their kids were playing together on the beach. So I'm grateful to the Bick and Carter kids for making this webinar happen. Dr. Carter is a biomedical engineer by training, and her research focuses on computationally modeling how DNA mutations in cancer impact intracellular biological processes. And today she's going to share with us her recent preprint entitled Interface-Guided Phenotyping of Coding Variants in the Transcription Factor RUNX1 with SEUSS. And I'm sure you all feel the same way that I do. I was really particularly interested in Dr. Carter's work because developing tools that can help us further classify variants of unknown significance are really critical for patients and their physicians and genetic counselors, because as you all know, it really leaves the healthcare team without sufficient data on how to base their management decisions, and can result in profound implications for patients who have hematologic malignancies and, of course, their families as well. So we're delighted to have you here with us today. And I should have also introduced Dr. Kivel Oztark, who is a postdoc in Dr. Carter's lab, and she'll introduce her as well, who's here as well to help with any questions. So looking forward to your presentation. Thank you again for being here, Dr. Carter.
Thank you for the kind introduction. I think you covered everything, so I'm just going to jump right in. So yeah, this is a new study for our lab. We are a computational biology group at UCSD that is interested in variant interpretation and precision medicine, especially for cancer. So this is a collaborative project with Prashant Mali, who's also here at UCSD in bioengineering and took care of all the experimental aspect of the talk. My postdoc Kivel is here, and she has done the bulk of the computational analyses. And Rebecca Panwala is a postdoc, a former postdoc with Prashant, who actually did all of the work with the cells and the experimental aspects. And then there'll be some other folks at the end that we'll acknowledge. But so first, I wanted to start with sort of a motivation that this sort of emerged from a way where we've been thinking about variant interpretation. There's a lot of tools out there that seek to predict the effect of missense mutations on the basis of protein level features, and sort of given a general score of whether a protein's function is going to be impacted or not. But proteins don't just exist in isolation. They work in these complex groups and pathways to mediate cellular behaviors in response to stimuli, et cetera. And that's what mutations are perturbing. And in cancer, missense mutations are one of the most common types of variant that we see in the tumor genome. And they're frustrating in that they can have a really broad array of effects on protein function. They can be completely destabilizing, they can have no effect at all, or they can have some intermediate effect. So we really want to think beyond just a simple score, trying to prioritize the functional effect of these. And if we think about them at the systems level, you can imagine a case like here on the right in the schematic, where this purple protein is sort of a hub in a protein-protein interaction network. It interacts with a number of different partners, and missense mutations affecting this protein could affect specific interactions and thus have effect that hits its direct interactors and their downstream targets. And so we can't really assume that every missense mutation is going to affect the system in the same way. So we've been trying to think about both computational and experimental ways to try to get at this, and that's what motivated the current study. Prashant has a technique called SEUSS that we're going to see used here that allows us to implement individual mutations and then profile them here with single cell technology. The focus of this project ended up being RUNX1, which is why I'm here today. And there were a number of reasons why we picked RUNX1. We know that it is a transcriptional master regulator, particularly for hematopoiesis, that it interacts with a number of different transcription factors to target it to DNA, where it is a pioneer factor and can activate transcription. It's primarily part of this core binding factor complex between a complex between itself and CBFB, which is crucial for its DNA binding affinity, but then a number of other partners are involved as well. And if we look at the mutational profile across cancers of RUNX1, we can see that a lot of the more recurrent cancer mutations are localized to the RUNT domain, which is the domain where it binds to DNA, but also where it binds to a number of its protein interaction partners, including CBFB. And as was mentioned before, it's commonly mutated in heme malignancies. The data, I think, that we've looked at the most has been TCGA AML, but we also see some mutations occasionally in breast cancer as well. So these were some of the reasons why we chose to focus on RUNX1. We thought if we were going to see any effect of missense mutations at a transcriptional scale, it would be in a master regulator, and this one is particularly relevant to a lot of cancers. To give an overview of the study design, we implemented a number of amino acid substitutions, and we included a set that we thought would have no effect on function, as well as a set that we were pretty confident would result in loss of function. as well as a wild type RUNX1 control and a complete loss of function GFP control. So the idea is that these variants need to be expressed in single cells, and then we can do scRNA-seq profiling to understand how different variants affect the transcriptional landscape. And then after we did our initial screen, we went and did some validation with bulk, and I'll go into more detail there. But because we're using a single cell RNA sequencing readout, we could not afford to do an exhaustive analysis of every possible amino acid substitution in the runt domain. We had to be clever in how we selected specific variants. And because we were interested in understanding the potential of variants to result in different, I guess, gene regulatory effects, we tried to pick amino acids that would affect interfaces between RUNX1 and its cofactors. So we first acquired a set of known interactors with experimental evidence of physical interfaces from the string database. And then we went to the PDB to look for co-crystal structures in order to map physical interactions. So looking at the proximity of residues in the two proteins to identify those amino acid positions that look like they are mediating the physical interaction. Unfortunately, there were not co-crystal structures for the majority of the binding partners in string, so we had to use their individual protein structures and a docking program, in this case PRISM, which can take the individual PDB structures and figure out the best way for those molecules to align, and then we can use, once again, the resulting model to figure out what the most likely interface positions are. And so here you can see a map of this. This is the runt domain proceeding along the 2D sequence. Here are the different binding partners. And if a block is colored black, then that indicates that we think this residue is mediating binding between, in this case, DNMT1 and RUNX1 in the runt domain. And then onto that, we overlaid information about the solvent accessibility of the position. The largest effect of an amino acid substitution on the stability of the protein using this FoldX energy field predictor. And then also the strongest possible pathogenic effect of an amino acid substitution here using the VEST classifier, which is actually a component of REVEL, if you're familiar with REVEL, which is what ClinVar uses. So now we have some idea of maybe these interfaces that these are using, and we can use this information about the likely functional effect of amino acid substitutions, the location at an interface, and et cetera, to try to pick individual substitutions that we thought would perturb these physical interactions. Once we had a set of interactions, then it was a matter of generating constructs by which we could express these in the cells. And here we chose to use the K562 cell line, which is actually a CML cell line. However, it has wild type RUNX1, which we thought was attractive because it seemed that RUNX1 itself probably wasn't a major player in the development of CML in that cell line. And so maybe we could just get a more unbiased idea of what RUNX1 might be doing, but you can imagine it will be important ultimately to look in different cell lines. The first thing we did was convert the cell line to have DOX-inducible RUNX1 knockdown so that we could get rid of endogenous RUNX1. So this is a construct that Rebecca designed that includes CRISPR guides, the CRISPR crab for inducible knockdown, and then here's the doxycycline inducible element. And then after we were sure that we could get pretty effective knockdown of RUNX1 in those cells, we went and transduced them with these viral vectors that had a construct with one of the RUNX1 variants that was hygromycin, with a hygromycin resistance element. So we could just select for cells that received one of these constructs, and then a variant barcode so that ultimately we could figure out which specific mutant that a cell received. So we grew these for 6 or 7 days under hygromycin to select for the ones that had successfully received a variant, and then we sent them for single cell RNA sequencing. And we did this with both the DOX-inducible knockdown of the endogenous RUNX1 as well as without, and did standard data cleaning for single cell RNA-seq analysis, filtering on gene counts, removing doublets, removing cells with a high percentage of mitochondrial DNA. We also removed cells that had more than one mutant barcode. And then there were 2 or 3 problematic variants where we either had fewer than 10 cells that harbored a variant, or I think there was one construct where instead of getting the missense mutation, a frame shift artifact was generated instead. But after all that filtering, we wound up with 112 variants in our library, and just the docs treated case, we had 20,878 cells. And you can see here in this plot sort of an idea of how many cells harbored variants. So this is across the entire library. And then you can see for wild type and negative control conditions, we tended to have somewhere around 100 cells. Whereas for the loss of function and positive control conditions, we tended to have more. And then the perturbations that we have no idea what their effect is going to be sort of span the range. So we found we could pretty effectively study the variant effects with fewer than 100 cells, but it was nice to have at least 100 for the vast majority of variants. So from here, after cleaning the data, we can generate a UMAP. This is after removing cell cycle effects. And here you can see unsupervised clustering of the single cells sort of suggested 3 groups. To get some intuition of what's going on here, we can overlay our labels from our controls as well as the sort of negative and positive mutation sets that we included. And you can see we sort of have this loss of function side and more of the wild type like and negative controls clustering over here. You can superimpose all the individual variants on this, but it gets really messy and hard to look at. So we tried to instead go back and get sort of average gene expression profiles across all cells harboring a specific variant, and then look at how the variants clustered. And so this is sort of a different low dimensional space into which we've mapped these variants. And you can see here that the wild type and negative controls are clustering over here, and our loss of function and positives are down on this end. If we repeat unsupervised clustering, it's suggesting once again 3 clusters. And since these are sort of separate from any of our wild type negative controls or loss of function controls, we sort of speculated these might be hypomorphic variants. So we're going to call them that and move forward with that label. And then if you map these back onto the original UMAP, you can see that these hypomorphic variants seem to be most concentrated somewhere at the boundary between in this UMAP between the wild type like and the loss of function. So this is how we've kind of labeled our variants, and now we can dig in a bit more to the single cell RNA sequencing profiles. To try to give them a functional score, we used Hotelling's t-squared statistic, which is a multivariable generalization of the t statistic. And we applied it to the top 20 principal components across the 2,000 most variable genes. And then we further measure a distance in this space relative to wild type and relative to loss of function. So this is kind of our transcriptional phenotype score that we can generate. And here we're plotting basically the distance to loss of function and the distance to wild type in the transcriptional space for each of the individual variants. And you can see that our loss of function like variants have very little difference. They're close to 0 in the loss of function space, but then they sort of span a spectrum of effects in the wild type space. And here are the hypomorphic variants that are kind of deviating both from the wild type and the loss of function like transcriptional space. We also did a fitness screen where we measured the abundance of mutant barcodes at days 7 and 14 relative to day 2. Here, we're going to calculate fitness as the abundance of a variant barcode at day 14 normalized to day 2, and we have 2 replicates, so you can see that the fitness was pretty reproducible, and that the wild type and loss of function had different extremes of fitness, with the hypomorphic generally falling in between. And I note the loss of function tended to have more cells associated with it and have more like a higher fitness score, which is consistent with the role of RUNX1 being a tumor suppressor in this particular cell line. Here we're now ordering the variants according to increasing T2 score, with the round circles being the wild type, so distance from wild type, and then the X's being the distance from loss of function. And you can see that the fitness trend correlates pretty well with this distance from wild type expression profile. And then we can look at how these mapped back to the predictions by FoldX and VEST. Just as a quick aside, we also just took a look at what happens when we do the knockdown of the endogenous RUNX1 with doxycycline versus not including that knockdown. And as you can see here, both for the distance to wild type and distance to loss of function, we still see an effect, but the effect size is smaller. So knocking down the endogenous RUNX1 really was helpful for getting, I guess, better resolution of the potential functional effects of these variants. So now we have these sort of groups of wild type-like variants, hypomorphic variants, and loss of function-like variants. We can start to look at the gene expression profiles to see if we can find some differences. And indeed, when we just look at the top 2,000 variable genes, and here each column is a variant, and we're looking at the mean expression profile across all cells for that variant, we kind of see this natural switch between the wild type and loss of function variants for different groups of genes, which are clustered along the y-axis here. In the hypomorphic set, we didn't want to just assume that all the variants had an identical effect on RUNX1 activity because they're potentially hitting binding sites with different interaction partners. So we further divided this into 3 putative groups based on the dendrogram here. And at the bottom, you can kind of see with this density plot where the centroid for those groups is falling within that UMAP. So they have sort of slightly shifted centers in this UMAP. And so yeah, so this is something that we're interested in exploring in this analysis, and I think we do see some evidence that there can be differences. But before we get into that, we can look at these different groups of genes and see functionally what is cohesive about them. And I think this is sort of more confirmatory than anything. We see that in these wild type-like cells, there's a lot of gene sets related to immune system function, positive regulation of T cells, proliferation, phagocytes, neutrophils, et cetera. When we look instead at the genes that are up in the loss of function light condition, we see genes associated with heme biosynthesis, angiogenesis, and regulation of the extracellular matrix, which aligned pretty well with what we saw in the literature. And then when we tried to look at these sets that maybe looked specifically different in the hypomorphic case, we saw some things associated with tau protein kinase activity and related to some neural aspects of RUNX1 regulation, like response to neural growth factor, which we found also some literature support for RUNX1 playing a role in aspects of neural function and development. In addition to just looking at the sets of genes that are expressed similarly, you can actually do something like this non-negative matrix factorization based assessment of gene expression patterns, which it tries to back out groups of genes that are expressed similarly in groups of cells and generates these patterns. And so you can get maybe a better fine-grained resolution to looking at sort of groups of cells that are doing similar things. And these basically showed very, very much the same that we're seeing in the wild type-like cells. More immune system function, and the loss of function more of the heme biosynthesis. And then in the hypomorphic, we were seeing maybe some evidence of some cell cycle effects and G1 senescence and interleukin signaling. We wanted to be more confident, though, that the single cell data was providing us with meaningful information. And so what we did is we picked these several of these hypomorphic variants and one of the loss of function-like variants, and we grew them out in bulk cultures in triplicate, and then did bulk RNA sequencing and bulk attack sequencing, and tried to see if we could get the same results when we have the variants in a bulk setting as opposed to across single cell where we have a lot of dropout and potential for artifacts. And actually, when we took the same top 2,000 variable genes from bulk RNA sequencing, we got very similar clustering of the variants. We got very similar relationships between the the T2 wild type score and the fitness activity, with increasing distance from wild type being associated with higher fitness scores once again. And when we do a PCA on those top 2,000 variable genes, the greatest axis of variance aligns with the wild type to loss of function axis. And then the second axis is sort of spreading out these individual variants. So we get the same basic intuition about the variants when we just use the bulk RNA sequencing. We also wanted to make sure that the effects that we were seeing were potentially due to perturbing interactions and not just destabilizing the protein itself. And so actually Nathan Payne and Dong Zhang's group here did some Western blotting of these selected variants as well. And this was sort of confirmatory that while sort of the loss of function variants looked like they were destabilizing, a lot of these hypomorphic variants had protein levels that were comparable to the wild type. And you can see sort of looking at the protein expression level versus the distance from wild type, we see that the loss of function variants here have the lowest protein expression, and wild types toward the high end, but then there's not so much correlation amongst the hypomorphic variants in terms of the functional effect versus the protein expression level. So we think that the single cell RNA is giving us a pretty good idea of what the differences between the mutations are, and does not solely reflect effects on protein abundance. To try to better interpret why these mutations might be resulting in different transcriptional profiles, we wanted to look at bulk attack sequencing. So for those selected variants, we grew cells out in bulk with expressing one single variant, and then did attack sequencing. And then we cleaned that up, called peaks, and then clustered the variants based on peaks. And when you cluster based on the attack peaks, you get, once again, very similar clustering to what we got based on those top 2,000 variable genes. And here is now a direct comparison of the PCA on the RNA sequencing versus the attack peaks. And once again, the largest axis of variance is this loss of function to wild type, and then we're getting some spread on the second axis of these variants, and maybe the attack peaks are giving us a little bit more spread even than the gene expression profiles are, to distinguish the individual variants. To try to study the regulatory impact of variants, what we did was we first called differentially expressed genes associated with hypomorphic variants. And what we were looking for specifically were genes that were up or down regulated relative to both the wild type and loss of function, as we thought these might be the most interesting specific effects for these hypomorphic variants. This is done with the 3 replicates that we had for each variant. And then for those genes that were differentially expressed, we tried to find attack peaks that would be associated with those, and that also had evidence of a RUNX1 binding site using Homer to look for RUNX1 binding motifs. And so if we found an attack peak with a RUNX1 binding site in the region near the transcriptional start site of a differentially expressed genes, that was of interest. But we also use this method Chrome-HMM by Jason Ernst, which we trained on publicly available K562 data from the ENCODE database to actually see if the attack peaks overlapped with regions that Chrome-HMM was predicting to be enhancers in the K562 cell line. So we had predicted enhancers from Chrome-HMM, promoters based on distance to the transcription start site, and then we're looking for attack peaks with RUNX1 binding sites that intersect those features. On the right, you can see here are the genes that are upregulated relative to the wild type and loss of function in the hypomorphic setting. And here we can see genes that are generally downregulated in these hypomorphic variants relative to the wild type loss of function. And the genes that are upregulated in the hypomorphic setting seem to be associated with interleukin signaling. Maybe there's potentially some UPR gene expression in there. And then in these ones that are downregulated in the context of the hypomorphic variant seem to be biased towards growth factor signaling, including FGF-R and IGF-1R. So yeah, these were the ones that we tried to focus on a bit when we looked at the regulatory aspects. And then just to give an example, this is for this variant G100V, and we have some visualizations we pulled out for genes where we saw different kind of relationships to the wild type and loss of function. So here with PTPN22, you can see the gene expression tracks on the top, where this gene is expressed in the wild type, not expressed in the loss of function control. But here in this G100V variant, it seems to be at an intermediate level. And then you can see there's a promoter here with a RUNX1 binding site, and we're seeing a peak in the TAC data for the wild type and G100V conditions and not in the loss of function condition. And similarly, here's a case in CXCL2 where we see higher expression of the gene in the variant condition relative to both the wild type and loss of function. And then in FGFR1, we're seeing lower expression in the G100V variant relative to wild type and loss of function. And so this maybe at this point somewhat hypothesis generating, but we could now go in and see if variants at some of these RUNX1 binding sites could actually modulate the expression of some of these genes. So finally, I wanted to look at mutations and how this information might help us study mutations in RUNX1. So initially, we were focused on cancer, so that's where we started. We collected missense mutations observed in tumors in the COSMIC database, as well as from the Munich Leukemia Laboratory, thanks to Torsten Haferlach and Stefan Hutter. And then we sort of looked first to see, was there any direct overlap between variants observed in tumors and the library of variants that we studied? And here you can see our library of 112 variants with their wild type, the difference from wild type score. So the loss of function here, the tall purple peaks. And you can see the frequency of these in COSMIC, so the number of times it was observed in a tumor reported in the COSMIC database on the bottom. And you can see how those align, but this is maybe a more practical plot where you can just see how the mutation frequency correlates with the functional score based on the transcriptome. And so you can see that definitely some of the recurrent mutations tend to have higher functional scores, although it's not a clear, there's a trend, but there's also cases where things that are highly recurrent are not necessarily the ones that are creating the most deviation from the wild type transcriptional score. Here's the similar plot for the MLL data set, right? We're seeing similar peaks emerge. Some are maybe more common in this data set than in COSMIC, but it's pretty similar. And then, so here is just showing that a few of these mutations, particularly the more frequent ones like 139 and 174, are actually here at the DNA binding domain of RUNX1. We also see some variants that affect the binding interface with CBFB. Definitely seems to be a general bias towards more functional mutations at the DNA binding site in general. This is only the set of variants that actually overlap with our library, though. So what we wanted to do was say, could we use the information we generated so far in these 112 variants and build a model that would allow us to predict for other variants in the runt domain what their likely effect on the transcriptional profile would be? And so here we have, we included in this a set of 79 variants, 41 wild type like. And then we grouped the 14 hypomorphic variants and 24 of the loss of function like variants into a positive class. And then we further split these variants into a training set and a test set just to see, is this going to work? So each of these variants was then characterized with a set of features. We took advantage of the SNVBox database, which has a bunch of pre-computed features for missense substitutions, including physicochemical properties, sequence conservation measures, some annotation of functional sites and structure-based predictions, et cetera. And then we asked, can we train our random forest to use this information to distinguish between the mutations that have no impact on transcription and those that have a functional impact on transcription. And then we evaluated our model on the held out 30 variants in the test set, and then compared also to predictions by VEST and FoldX. And actually, it seemed like it's doing a pretty good job at predicting for the held out set what the transcriptional impact is going to be. This is an ROC curve, so perfect performance would be on the top left here and the top right on the precision recall curve over here. And we can sort of summarize performance with this area under the curve metric. And so it outperforms just general effects on protein stability or pathogenicity prediction based on all genes, maybe suggesting that a RUNX1 specific classifier has some advantage in this context. So first we applied this to cancer mutations. We had an additional 657 runt domain variants in tumors, and we can now predict what their effect on transcription is going to be. Here's the these are the effects versus the frequency of these mutations in COSMIC and MLL databases, with the assumption that things that have a higher functional impact are probably more likely positively selected and should be more recurrent in cancer. And that's exactly what we see. The higher frequency mutations tend to have higher functional scores predicted by the model. And we also see these mutations are, the functional mutations are enriched in these data sets relative to something like NOMAD, which is not a cancer data set, it's just average, a data set of individuals that are generally thought to be healthy. Finally, thanks to a helpful suggestion from one of our reviewers, we applied this to variants in ClinVar. And so there was a small amount of overlap between the variants we probed with the single cell RNA sequencing and ClinVar. Namely, there were 24 variants, of which 8 were either benign or pathogenic. There's only 1 benign, the others were pathogenic. And then 16 were variants of uncertain significance. And here we can see that the benign variant was very close to the wild type, and then the pathogenic and variants here coincided with our loss of function and hypomorphic predictions, and started to have transcriptional profiles that were pretty different from the wild type. And when we looked at these 16 variants of uncertain significance that we'd measured transcriptional profiles for, we are able to sort of identify some that look like they should have a potentially functional impact on the transcriptional landscape versus some that look like they should be very similar to the wild type protein. And then for an additional 124 variants in ClinVar, we had not directly measured the transcriptome, but we can take this random forest classifier we built and apply it to those variants and look at how the scores predicted by that classifier play out. And here, once again, we had 1 benign variant, which got scored as being one of the more wild type like mutations. The pathogenic variants were mostly scored as being functional. This cutoff here at 0.5 is somewhat arbitrary, and it's debatable whether that's the best possible cutoff, but we didn't want to overfit the data. But you can see that some of these pathogenic variants are sort of borderline with that prediction. And then this is, we see that this once again splits VUSs, and a lot of the VUSs are being predicted as likely functional. So maybe this would be a great place to start a new iteration if we wanted to look at additional variants and what their functional effect might be. Okay, so just to wrap up, we used interface guided mutagenesis and a pooled overexpression single cell RNA-seq assay. And what we were trying to do was identify ideally hypomorphic variants, and we did find a few of those. We were able to validate that the single cell approach is working pretty well by comparing it to bulk RNA sequencing for a subset of mutations. And for the hypomorphic variants, we showed that their effects are not just due to decreased abundance of protein, but they seem to maybe actually be perturbing RUNX1's probably binding and regulation of his downstream targets. We were able, because we use K562 cells, to take advantage of a lot of data in the ENCODE database. And then we added our own bulk attack-seq data for specific variants, trying to get at potential differences in gene regulation attributable to these mutations. And then finally used machine learning on this sort of phenotypes we got from the single-cell RNA sequencing readouts, which seem to work pretty well for predicting variant effects. But I think there's a lot of variants that we didn't sequence directly, so there's potential to go back and try to build new libraries from variants that we now think might be important. I'd be very curious what this group thinks about cell lines that we should potentially look at these in. And then another thing that we're working on right now is to try to do affinity purification mass spectrometry for some of these variants to see if we can actually detect differences in the transcriptional activator complex in terms of which proteins are involved when we have some of these hypomorphic variants. And maybe that will coincide with some of the differences in peaks that we see in the attack data in terms of gene regulation. So, of course, this work wouldn't have been possible without fantastic collaborators and an amazing team. And as you know, Kibble is here with us today, and she did the bulk of the computational analysis. Rebecca Panwala and Prashant's group did the bulk of the experiments, but was also supported by Gina Sheen, a master's student who's now graduated. She's here next to Kibble. And if you're interested in the paper, you can scan this to go to the bioRxiv preprint. And I guess thank you very much for your attention. I would love to hear questions or suggestions.
Thank you so much, Dr. Carter. That was awesome. That's a lot of work. Congrats, Kibble. Please go ahead and ask any questions via the Q&A icon. We are open to having a discussion, and I'm also interested to hear what people think about what other cell lines should be tested. I know there's a lot of controversy in the literature about whether RUNX1 is a tumor suppressor or an oncogene, and it does seem to be cell specific. But I think that'd be really interesting to see what happens when you test this in a totally different cell line, but also in another hematologically based cell line. So open to hearing what folks think. One of the questions I had for you is in the last slide, you showed that you had some pathogenic variants that were just below your arbitrary borderline cutoff. I was curious, when you looked at those variants, was there anything unique about those variants and where they reside in the protein domain?
That's a great question. I'm not sure we've really looked at the ClinVar variants specifically in terms of where they're falling in the runt domain there, or whether they show the same enrichment for the DNA binding effects, right?
Yeah.
That's something we absolutely should do.
Yeah, that's a good suggestion.
Great. We have a comment from one of our attendees. You should consider testing in a +21 cell line. I think that makes sense, right? Because of RUNX1 being on chromosome 21. Good suggestion. Another question here. It might be hard to say, but do you think the predictions would be better in a hematopoietic stem cell versus a K562 CML cell line? The gene expression changes may be different. Issue may be that CD34 positive hematopoietic stem cells will differentiate over time. I think it is an interesting question because you are doing this at the single cell level, so you would be able to get those cells. What are your thoughts?
Yeah, no, I think a stem cell line could be quite interesting. I'm not sure. I guess like for a cancer setting, right? You think, I don't know, any place where we have like wild type RUNX1 for starters, I think would be more important. I'm worried with some of the cancer cell lines that the development of the cell line has required some kind of perturbation to RUNX1 to begin with, and then that will color whatever readout that we get. So I think maybe the stem cell would be less prone to that. I don't have a lot of experience with the challenges of working with stem cells in the dish, though. But I believe Prashant does quite a bit, so he might be really interested in trying that.
Yeah, Absolutely. We do have some folks in our community that have set up systems with CD34s and then RUNX1 mutations. So could be worthwhile. Let's see, another question here. Regarding the fitness of the different mutants, what is meant by abundance of the mutant barcodes? Are the cells not completely edited, or is it an absolute number of cells?
I think the cells, we really target to get them just one single mutant, but then when we look in like the population of cells after they've been transduced, we can count the number of cells with each of the barcodes. And then over time, like 14 days, they're allowed to grow, and the barcodes that are associated with, I guess, better growth of the cells will then at the last time point have increased in abundance, whereas barcodes that are disadvantageous will maybe have those cells will have fallen behind. They're not proliferating as fast, or maybe they're dying. And so we can sort of see which cells kind of grew out and which ones didn't, which barcodes were associated with that. And that tells us which mutations gave a fitness advantage, if that makes sense.
Got it. I actually had a follow-up question. One of the things that struck me when you were showing in the very beginning the difference between the clustering of the different mutations, when you clustered based on loss of function, wild type, and hypomorphic. Across all those figures, I remember hypomorphic kind of being in the middle with fitness, et cetera. But then when you started looking at transcriptional changes, they ended up being the more extreme. I just found that really interesting. Do you think that it's because the hypomorphic variants are having totally different binding partners than, whereas in the loss of function setting, you're basically just a wild type. It's a wild type situation. I guess I just was struck by loss of function having this huge fitness advantage, but then when you looked at transcriptionally, they were closer to a wild type.
Yeah, I don't know if it's completely fair to argue that all of the wild type like mutations are exactly like wild type and all of the loss of functions are exactly like loss of function.
Fair
But
enough.
I guess we speculated that destabilizing things would generally have the same transcriptional readout, right? Whereas the hypomorphic, at first we thought maybe they were just sort of some kind of intermediate. Maybe they just have like weaker binding affinity or something, and you're just getting sort of a weaker version of the wild type profile. But what we were really interested to see was that it did look like there were some genes that were being expressed differently relative to both conditions. And that's when we thought, okay, maybe these really are perturbing in some way the transcriptional complex and changing how RUNX1 is locating to the DNA. And maybe through that, we're getting these kind of deviant transcriptional profiles. This is the first protein that we've tried this on. And as a master regulator, I can imagine that RUNX1 function is probably evolutionarily very constrained. So I don't know, maybe we shouldn't expect there to be a ton of hypomorphic alleles, although I think there's a lot more space to explore and potential to optimize how we're picking variants that might perturb the complex formation. And maybe we would see like more radical hypomorphic landscapes for other genes if we were to look, but certainly in our study, it looked like the majority of mutations either were complete loss of function or wild type.
Yeah, yeah. A comment, it looks like, regarding cell lines and what to use next. One is on potentially maybe using male and female induced pluripotent stem cells and differentiating them to different myeloid fates. But the follow-up comment was that probably would be expensive given the cost of cytokines, et cetera. But another idea. It looks like I've got a question in this one. Really wonderful work. Do you think your classifier is ready? This is a great question. Is ready for clinical implementation for deciphering VUSs? And if not, what would be required to move forward towards a clinically useful classifier?
That's a great question. I think it already it looks like it's providing some interesting information. One of the things that I'm personally really interested in, though, is how genetic background plays with these things. And right now we're only looking at K562. And as you mentioned, it could be that RUNX mutations are gain of function in certain contexts rather than loss of function. And also it's quite possible that there are secondary variants that will modify the effects of these RUNX variants. And so I think, it certainly would seem to provide some additional information for some VOSs that I think maybe could be helpful, but I'm not sure how you get it past a bar to say that it's like safe to completely follow that assumption. I guess the ClinVar has done a ton of work though on trying to get these likely pathogenic designations from classifiers and have some criteria for that. Might be interesting to see how we stack against their current criteria, and maybe that's the path for trying to see how this could be moved in that direction. But at worst, we could take some of the predictions where it says things are looking very much like they're pathogenic, and test some of those in additional systems, maybe to boost confidence.
Great, absolutely. We could connect you with the MMV group. They're the expert curators. The APMS follow-up seems super interesting. Do motif analyses of the genes that change expression or activation suggest high priority interaction candidates as opposed to co-binding due to motif proximity?
That's a great suggestion. I think we could do more on trying to look for other motifs related to the binding sites or the differentially expressed genes that we have so far. But I think a lot of the partners don't always directly bind the DNA themselves. I think we found there were a subset that did, but not all of them. So I think that's definitely a place where there's room to do a bit more analysis, and hopefully when we're working on growing out the cells to try this APMS experiment, it would be really rewarding if we can see some differences and then see that the binding sites for some of those proteins are reflected in the attack peak data with the corresponding mutations.
Awesome. I'm going to pause to see if anyone has any other questions. And in the meantime, congratulate you again, because it's some really great work, and appreciate the fact that you're working on these tools. Oh, got another question. Have you looked at the variants at the C-terminal region?
We stuck to the runt domain so far. We could expand outside of it. Definitely any place where there's physical interactions with other proteins would be really interesting. I think we can apply our classifier to predict effects, but I would have even less confidence about how well it's generalizing outside of the runt domain. But we can at least already make predictions potentially, and then use that to maybe try out a library on the C-terminal domain.
Can I ask actually why are you less confident if you're outside the runt domain?
I guess I would assume that that locally there might be some shared information across the sorts of amino acid substitutions that are perturbing function, and that maybe as you get more distal from that it's possible that there's different information, right, that we're not capturing because we didn't probe what's important in that that region of the protein. It's possible it could generalize quite well, but it's also possible that there could be some different optimization of sequences in the regions for various functions, and then it has slightly different I guess benefit or damage by specific substitutions, right?
Yeah,
Maybe you need more of a certain charge in the DNA binding region, but maybe not in the C-terminal domain, for example.
sure. Okay. Understood. All right. I think I'm going to give it one more second to see if anyone has any other questions. All right. Well, I appreciate you both taking the time and sharing this presentation with us, and we'll share it with those who, especially those who registered and couldn't make it. Yeah, keep in touch. And I know that there's a whole community here that is, working on RUNX1 and super interested in the work that you did. And if there is opportunity to, if you're interested to expand out and test your platform on C-terminal, on the C-terminal region variants, for example, I think that would be of interest, because we have a lot of patients who come and have VOSs and are wondering, do they have the disease? Do they not? It's pretty challenging for physicians and individuals. So anyways, thank you very much. Have a great rest of your day. Take care.
Yeah. Thanks everyone for the great suggestions and questions. And another shout out to Alex and his daughter for getting us connected with this group. And thanks so much for the opportunity today.
Yeah, absolutely. Take care. Bye.
Bye.
Bye, everyone. Thank you.